Thank you very much, Professor Kazab, for this kind introduction, and thank you all for being here at this wonderful meeting organized by Dr. Charles Balch and Dr. Ning Liao in this beautiful place. This picture there is the view from my room. Which is very nice. And I think the sun had come out a little bit last night. These are my disclosures with some images that we enjoyed over the last few days at Chimelong in different cities in mainland China. And as Dr. Kazab mentioned, I'm really known in the field for a couple of accomplishments leading up to some of my current efforts in the field of precision oncology. I was very fortunate decades ago to work in the laboratory of Bert Vogelstein at Johns Hopkins. It's always important to remember our mentors who have helped us and shaped our careers. And at the time, we discovered P21 as a P53 target gene. And this taught us about the cellular response to DNA damage so that when cells arrest and stop dividing, they have time to repair damage and avoid turning into a cancer. And this was followed by development of small molecules such as CDK4/6 inhibitors, widely used in ER/PR positive breast cancers, and coming along in other tumors. Now, when I started my laboratory at the University of Pennsylvania, I was interested in cell death. Why? Because I'm a medical oncologist, and I understood 30 years ago that we want to kill cancer cells. If we kill the cells, they're not there anymore. And so that seemed like a good approach to making a difference in the field. And we ended up connecting the major tumor suppressor to the innate immune system through direct regulation of a TRAIL Death Receptor 5. This was a connection between tumor suppression and the innate immune system. And over the last 2 decades, I've been thinking and trying to find ways of addressing this or using this knowledge to develop new treatments to help patients with cancer. And as you can see with this background in cell cycle and cell death, this goes across tumor types. But of course, we have to understand the differences between the tumor types as well as the differences in our patients in their ability to mount anti-tumor responses. The cell death pathways are complex, and they occur within the cell as well as outside the cell. They involve the immune system and engage families of different receptors. And I think everyone who treats cancer understands the multitude of resistance mechanisms that allow evasion of our therapeutics. And so this was a major focus of our research. And as I said, so back in 1997, we reported the discovery of this innate immune receptor regulated by P53. And by 2007, we became interested in screening for drugs to boost this innate immune system in a P53 independent manner because P53 is mutated in most cancers. And we discovered a molecule, TIC10, trail-inducing compound number 10, also became known as ONC201, that entered clinical trials by 2014. And by 2017, it was becoming clear that this molecule has activity in aggressive diffuse gliomas in the middle of the brain, often unresectable, not particularly responsive to the only standard of care therapy of radiation, and they certainly are known to unmethylate MGMT and so are resistant to temozolomide. And this drug seemed to have activity specific to this patient population. And the drug was approved as the first ever treatment for H3K27M mutant diffuse midline glioma on August 6, 2025. And, I started out not planning to develop a drug to treat brain cancer. The drug does have activity in other tumors like neuroendocrine tumors, prostate and endometrial cancer, but those clinical trials and path to approval are ahead of us. So, I'm trying to figure out how do we make this class of drugs work better? What about analogs? Anyways, I can talk for hours about this, but I won't because I'm here to talk about the WIN Consortium. And again, I'd like to thank Dr. Balch for bringing us all together and for driving home the point that we spend time together, we get to know each other, and hopefully we collaborate and make a difference for our patients. And some of these interactions are less formal and fun, but do have the intended impact. So I'd like to provide you an update about the WIN Consortium, its activities, various projects that are underway, and where the consortium is going and in the future. So the WIN Consortium was formed on the premise that consortium members can accomplish together what no single institution or group of patients or individuals can do alone. And it has become clear over the last 16 years that precision oncology really is the future of oncology. It's what any of us would want for ourselves, our loved ones, or our patients. And often, I very much appreciate the theme of this meeting and ISOPT. We really do want to prevent cancer, but sadly in 2026, many of our patients are not diagnosed early. They present with advanced disease, often having exhausted standard of care, and maybe there is no perfect clinical trial for them. And so precision oncology offers a way to bring to the patients and their clinicians ideas of potentially effective combination therapies after discussion by an international molecular tumor board. So the WIN Consortium was founded by Dr. John Mendelsohn, the late president of MD Anderson, Dr. Thomas Terse, the late director of Gustave Roussy. I very much appreciated meeting Dr. Egermont for the first time today. Enjoyed his talk and the impact of neoadjuvant therapy across tumor types. And other leaders in oncology have had associations with the WIN Consortium, including Dr. Tabernero, Dr. Richard Silsky, who became the chair when John Mendelsohn became ill and sadly passed away from a brain tumor. And I was elected the chair in December of 23 and was reelected in December of 25. Currently, the WIN Consortium has 38 members from 19 countries and 5 continents with access to over 5 million patients. And if you look carefully on the right side, you will see that ISOPT recently became a member of the WIN Consortium, Dr. Balch, and Dr. Weitzel have graciously agreed to co-chair a committee on screening and precision prevention, and we look forward to seeing where those efforts lead us as we collaborate and try to have impact around the world. This is the global footprint of member institutions. Our executive committee is there. We recently have now two vice chairs of the WIN Consortium, Dr. Tsiveridou from MD Anderson and Dr. Lamai from Sir H.N. Reliance in Mumbai, India. Dr. Rizal Kurzrock, the chief medical officer, is one of the WIN Consortium, is one of the premier clinical trialists in the world and champion in the field of precision oncology. Our directorate has global membership, including now representatives of patient advocacy groups. And our scientific advisory board, chaired by Professor Yves Lussier, now has two vice chairs, Professor Liang Cheng and Professor Roy Herbst, representing pathology and oncology clinical trials, as well as global leaders in the field of oncology. And I'm not going to read all their names, but you can see the pictures from various associations and cooperative groups and areas of expertise. Dr. Balch is a member of our scientific advisory board, as is Dr. Grelo from ASCO. WIN has had a number of standing committees. We have an annual symposium, so we have a symposium committee. We have a publications committee. We have an international molecular tumor board that I'll tell you about, a membership committee. And we have activated a number of committees over the last couple of years. The first one we activated was a data science committee. We have a rare cancers committee, GI committee, a GU committee, thoracic committee, with leaders and co-chairs from around the world, including Dr. Rugo, who's here at the meeting, and Dr. Corigliano, who graciously agreed to co-chair this committee, along with other colleagues, including from Fudan University. A number of other committees, and it's not just disease committees. It's really, we have an interest in novel agents. Regulatory issues are very important because the field faces a number of challenges in terms of coverage for drugs and oversight of this emerging part of oncology that is not yet quite mainstream, but will eventually get there. And at the bottom, you can see our Precision Oncology Screening and Detection Committee with the co-chairs that I mentioned. Now, we do have an International Molecular Tumor Board that meets regularly to discuss very challenging cases and brings together the experts from around the world. I am pretty active on social media, and I report on these molecular tumor boards. And we have taken an effort to publish the case reports with the decision that the thought process at this molecular tumor board is very important for our colleagues around the world. Many of these patients do not have clinical outcomes. That would be a future direction and a project for anyone who is interested, because the consent forms allow that. But the thought process of coming up with options and prioritizing them, and the types of questions that are before us include what do we do in the adjuvant setting? How do I keep my tumor from coming back? And it's interesting after hearing Dr. Egermon's presentation, we end up having conversations about cancer vaccines and CAR T therapy, immunotherapy. So it'll be interesting and important to see how those things play out in the setting of patients whose tumor responds to neoadjuvant immunotherapy. But of course, not everyone will respond in the same way. And again, the field of precision oncology offers an opportunity to determine who benefits and how to help them better. Some of the other questions come up where patients are in remission and want to know what to do when their disease comes back. This is remission from advanced disease or recurring disease with multiple alterations, molecular alterations, not just mutations, but other proteomic changes and other information, including results from AI algorithms that we are looking at these days around the world. This year, we started an international fellowship in precision oncology, co-led by myself and Dr. Rizal Kurzrock. And we have 3 fellows from around the world who have joined. And as has been demonstrated in other societies, fellows can drive progress in certain areas as they advance their careers. And the WIN Consortium serves as a wonderful venue for their training and support as they accomplish their objectives. We invite new members in the WIN Consortium, and some of the what members can expect is to be involved in the strategic direction of the consortium, to collaborate with pharma foundations for support of directions, innovative directions, bringing new concepts to disease committees, attending WIN symposia, and being involved in education and mentoring activities. So I'm going to give a quick overview of some of our ongoing past and future projects. The Winter trial was the first to use transcriptomics in addition to genomics to select therapy for patients with cancer across tumor types. And this trial enrolled 303 patients who were heavily pretreated, where a quarter of the patients had more than 5 lines of therapy. And the results were published in Nature Medicine in 2019. And the result that you see in the right is overall survival as a function of matching score of combination therapies for a given patient's tumor. So this matching score has emerged as a predictor of overall survival and benefit in the field of precision oncology. And of course, in 2019, we had far fewer drugs and drug combinations at that point. The spring trial was a smaller trial in lung cancer, testing a novel combination, and this was published. There is a database called OncoAid that provides access to information about drugs, and you can go to the website and learn more about it. It was updated recently. We do have an active study with Tempus. This is looking at ctDNA prospectively in about 1,000 patients with non-small cell lung cancer at different stages. And a similar study is coming online in head and neck cancer known as the Phoenix study. We have recently submitted a grant application pursuing a trial of imipridones like ONC2 in combination with darolutamide in castrate-resistant prostate cancer, and recently submitted an application for recurrent H3K27M mutated diffuse gliomas. This is a schema that is not only intended to provide new information for how to treat these patients better, but also what to do at progression using the molecular tumor board and all of the available modalities to the extent that they apply for specific patients. We've also submitted a number of studies that have not been funded, and if you see anything here, please feel free to reach out if you think there's a way we can collaborate. But this was a sarcoma precision oncology trial. This was a pancreatic neuroendocrine tumor trial. This one is particularly relevant across the world, which is a study that we submitted to the NIH entitled Mutations Across Borders. And I do think there's a lot to learn about different drivers around the world in the context of co-mutations and environmental factors that are different in different places, as well as how treatment plays out with all of the limitations and challenges that are faced in different parts of the world. So this sort of study does need to be done at some point through collaboration around the world. We've also submitted to Cancer Grand Challenges, looking at approaches using synthetic lethality. And this one, AIEE, is particularly relevant and important as a global trial. It's artificial intelligence for enrichment and equity. The artificial intelligence part involves digital pathology, and in this particular schema, WIN has been collaborating with Caris Life Sciences that have developed algorithms to enrich for specific driver mutations from H&E slides that can be uploaded from anywhere in the world. So the enrichment part is that. The equity part is, as has mentioned by others at this meeting, has to do with fewer tests being done on tumors and testing tumors that are much more likely to have driver mutations so the patients can get the correct treatments. This trial is currently at the stage of discussions with pharma companies. And so take a look at the different arms. And if you have connections or interest, whether it's institutional or multi-institutional, we would be very happy to work with you and collaborate. And these types of trials bring in the latest advances in precision oncology, including functional precision medicine. I will say, if there are pharma colleagues listening, the N of 1 precision oncology phase 1/2 clinical trial design provides the most efficient investigational path to identifying safety and efficacy signals for new drugs and new indications for approved drugs. And whether you're at an institution or a pharma company, you know what's exciting. We've heard about the different modalities and approaches. You should ask yourself, what's in my portfolio for precision oncology? This is the general paradigm of those N of 1 trials that can start at the neoadjuvant setting or later, collect data with liquid biopsies and tumor biopsies and imaging, involve various AI algorithms, learn from them, and advance our knowledge and improve care for patients with different tumor types. The WIN Symposium is very active. Last year, we had Bill Kalin as our keynote speaker speaker. Our next WIN Symposium is October 2 to 3 in beautiful Rhode Island, where I live. We have a lot of water, and I would invite anyone here to consider attending this wonderful meeting. You could see some of the highlights of speakers, including Nobel laureate Tom Sudhoff from Stanford, Rick Pazder, previously at the FDA, and Keith Flaherty, the president of AACR. And there are many, many more, including speakers from around the world, including Dr. Balch. So I'm going to wrap up here with the Data Science Committee that I mentioned earlier. My challenge to the Data Science Committee was don't just come up with the challenges. So you could read the challenges that are there. What are the opportunities? And you could see what the opportunities are. And I've touched upon some of them in my presentation, but there are tremendous opportunities for collaboration around the world in the area of mining data, both retrospectively and prospectively. So basically, this is my last slide of where we are in the WIN Consortium, developing collaborations, submitting grants to support clinical trials. We're very actively publishing, including from the Molecular Tumor Board, and we do have an educational mission. We have the WIN Fellowship, and I think the disease committees are very welcoming to colleagues at different career stages and can be very impactful to allow people to network and work on career development and advance the field of precision oncology. So thank you for your attention.